Yes, good afternoon. KUTV, Elimu Live. I'm your host today, MUCHIRI Thomas. For today's lesson, a biology lesson, we are going to dwell on one of the major topics in biology, that is nutrition, which is the last topic in Form 1. And briefly, the brief introduction of this lesson, maybe you're going to start by introducing biology. From the word biology comes from two Greek terms. One, bios, that means life, and logi, that means study. So biology is the study of living organisms. Is a study of living organisms. And as living organisms usually have similar characteristics. For example, we have reproduction. All the living organisms have the following characteristics. One is that they reproduce. They also need food, nutrition. There is respiration. There is gaseous exchange. Living organisms move, so there is movement. They also respond to changes, so there is irritability. And you find that most of these characteristics of living organisms, they're the ones that comprises of the high school syllabus. Reproduction is the third topic in Form 3. Nutrition, the last topic in Form 1. Respiration, the second topic in Form 2. Gaseous exchange, the second topic in Form 2. There's movement in Form 4. Irritability also in Form 4. So our lesson today, we are going to dwell on this major topic, nutrition. And when you talk of nutrition, we are going to start by defining the term nutrition. Nutrition is a process by which living organisms acquire and assimilate food. So, living organisms acquire and assimilate food. We have two terms here. We have to acquire and to assimilate. The new term here could be to assimilate. The other name for to assimilate is to use or to utilize. Now, nutrition, we have two modes of nutrition. So, modes of nutrition. So basically we have two main modes of nutrition. The first one is modes of nutrition. The first one is heterotrophism. And this heterotrophism, this is where we have living organisms feeding on already complex food materials. And those organisms that exhibit this mode of nutrition, they are known as heterotrophs. Heterotrophs. They are known as heterotrophs. So they feed on both plants and animal tissues. In heterotrophism, we have 4 types. One, we have holozoic. We have saprotrophism. We have parasitism. And finally, we have symbiosis. So we have holozoic, we have saprotrophism, we have parasitism, and we have symbiosis. Now, the first one, let's start with this one. So this holozoic, this is where we find that living organisms, they ingest food. Ingestion is the introduction of food into the mouth. That's what you call ingestion. Then this food is there's chewing and grinding, what you call mastication. Then we have the chemical digestion by enzymes, and finally the digested food is absorbed into the bloodstream. Then for the undigestible and indigestible food materials, these 2 terms here, we have 2 terms here. The undigested, and indigestible food materials. This food that can be digested, but due maybe to possibly lack of due to poor mastication, the food was not well digested, was not mechanically broken down. But for the indigestible food materials, these are the hard food substances. For example, some of the seeds of the fruits that cannot be digested. And these 2, they are the ones that are removed into the alimentary canal, and we call that process egestion, the removal of undigested and indigestible food materials. From there we go to the second type of heterotrophism, which is saprotrophism. Saprotrophism, this one, living organisms, they feed on dead, decaying organic matters. These organisms, these type of heterotrophism organisms feed on dead, decaying organic matters. Then we have parasitism. This one, we have the organisms. There are certain organisms that depend on the other. The one that is depended on is called the host, and the one that depends on the other one is called the parasite. Then we have the last one there, symbiosis. Symbiosis, this is where we have the mutual benefits between 2 living organisms. For example, we have the rhizobium bacteria that are hosted by the legumes. They are able to convert atmospheric nitrogen into nitrates, and also they benefit from the plant by shelter. So the 2 living organisms benefit from each other. So the other type of mode of nutrition, is autotrophism. Autotrophism, for those organisms that exhibit this mode of nutrition, they are known as autotrophs. Autotrophs. Most of the students here, when you talk of organisms making their own food, we have that notion that only plants that can make their own food. So for the autotrophs, yes, we have green plants. Green plants manufacture food. We also have green algae. Also, it is able to make its own food. And finally, some bacteria. Some bacteria also are able to make food. So when you talk of autotrophism, we not necessarily talk about plants because it's only the first one there, the green plants that belongs to kingdom Plantae, algal belongs to kingdom Monera, and some bacteria, they also belong to Monera. So we have three other kingdoms that are not represented. That is kingdom Animalia, kingdom Protoctista, and Fungal. Those three, they are not represented in this mode of nutrition. So we go first to definition of autotrophism. Autotrophism. And autotrophism, we say that this is a mode of nutrition where living organisms manufacture complex food materials from simple substances. Living organisms, autotrophism, we say that living organisms manufacture complex food materials from simple substances. Maybe here we have considered the living organisms that you're talking about. We have the green algal, the plants, and some bacteria. Then these complex food materials that are produced, the end products of photosynthesis, which is at the autotrophism. We have starch, we have proteins, and you have lipids. So these are some of the complex food materials that are manufactured from simple substances. So these are complex food materials. And these complex food materials, where do they come from? So in this process of photosynthesis, we have requirements. We have the raw materials that are required in this process, and we also have the conditions that are necessary for this process to take place. So for the raw materials that are required, raw materials required in the process of photosynthesis, there are two. One, we have water, and we have carbon 4 oxide. We have water and carbon 4 oxide. Then for the conditions necessary, for photosynthesis to take place, we also have two. Only those plants that have got chloroplast, and chloroplast is the one that is that contains the green pigmentation, that is the chlorophyll. We have so they are the ones that you call photosynthetic cells, and the conditions necessary here, we have chlorophyll molecules, we have the chlorophyll, and secondly, sunlight is required. Sunlight is also a condition that is necessary in this process of photosynthesis. From there, maybe you're going to look at the process itself, the process of photosynthesis. Photosynthesis. Photosynthesis, we say that this process is a process by which green plants manufacture complex food materials from simple substances. when you're defining the term photosynthesis, the difference between autotrophism and photosynthesis is the fact that for the photosynthesis is only the green plants. But when you talk of autotrophism, it involves kingdom Monera. So we talk of living organisms, we cannot talk of green plants. So photosynthesis, it occurs in two stages. We say that occurs in two stages. The first one is a light stage. Then we have light stage, then we have the dark stage. We have the dark stage. Now the light stage takes place, or it occurs in the grana. Light stage occurs in the grana, and in the grana we have this condition, these chlorophyll molecules. So what is the role of these chlorophyll molecules? So the chlorophyll molecules absorb sunlight energy or trap sunlight energy. Chlorophyll molecules traps sunlight energy. After trapping this sunlight energy, what is the role of this sunlight energy? This sunlight energy, it is the one that is going to split the water molecules. It is the one that is going to split the water molecules to hydrogen ions and the oxygen. It is the one that is going to split the water molecules. It's the one that is going to split the water molecules to hydrogen ions and oxygen gas. So we have now the sunlight energy splitting the water molecules being split by sunlight energy to hydrogen ions and oxygen atoms. This process is known as photolysis. This process is known as this process is known as photolysis. It's known as photolysis. And in this process, there is production of energy. So energy is produced. Energy is produced in the form of adenosine triphosphate, what we call ATP. It is good to be noted that all these abbreviations that we have possibly have used here, for example, this one, water being split to hydrogen ions and oxygen. These symbols, they are not accepted in biology. Is only one that is accepted, the one for the DCPIP, the one we test for ascorbic acid, or what we call vitamin C. This is the only one that is accepted. Now that is all about the light stage. So as we started here, we said that autotrophism, we have that definition: living organisms manufacturing complex food materials. from simple substances, and we have seen that the complex food materials that we're talking about, we're talking of starch, we're talking of lipids, proteins, because those are the end products of photosynthesis. And for that photosynthesis to take place, we have simple substances that are being used. So we have water and carbon dioxide that are used as the raw materials. And conditions necessary for this process to take place, we have sunlight energy, and you also have chlorophyll molecules. Maybe somebody may ask the difference between the difference between conditions necessary for photosynthesis to take place and also the raw materials that are required. For the conditions, for example, when you're baking a cake, you may require a flour, we may require sugar, water. All those are raw materials that are required. But as well, you may require heat or source of heat, which as you take your cake, it will not be part of whatever you're going to consume. So it is only a condition that is necessary for that cake to be baked. In this case, our chlorophyll molecules and our sunlight serve the same as that heat, but not as a raw material. There's something else here that needs to be noted. When you talk of autotrophism, yes, we have talked about photosynthesis, but we have two types of autotrophism. One is autotrophism. The second one is chemosynthesis. Chemosynthesis. These are the two types of autotrophism. What's the difference between these two? This one requires, from the word photosynthesis, photo means light, synthesis means to make. So for photosynthesis, there are these living organisms that are using light to make their own food, hence the term photosynthesis. But for the chemosynthesis, so this one we have said that is getting the energy from sunlight. For the chemosynthesis, the energy that is required in this process of by which these organisms use this type of autotrophism to make food, they get energy from oxidation of chemicals. So these are the two types of autotrophism. And now from there, let's go to our second stage of photosynthesis, which is the dark stage. The dark stage, it takes place in the stroma. Remember, we have said that photosynthesis takes place in what we call the photosynthetic cells. Photosynthetic cells. We have in plants only 3 different types of cells that have these organelle known as chloroplast, and this chloroplast is the one that contains that chlorophyll. So one of the cells that manufacture food for the plants, one is the guard cell. We have the guard cells, and remember, guard cell is just but an epidermal cell, but it's specialized. Why is it specialized? Because it has got chloroplast organelle. It has got organelle, this organelle that we are calling chloroplast. This chloroplast is the one that has got what? Chlorophyll. This chlorophyll How is it used in this process of photosynthesis? It is the one that is trapping sunlight energy so that it splits the water molecules to hydrogen ions and oxygen gas. What we call photolysis. So we have the guard cells. The other one we have the palisade. The palisade cells. These ones, they are found after the upper epidermis. So on the internal structure of a leaf, we have the upper cuticle, upper epidermis, then the palisade mesophyll cells. Then we have the spogy mesophyll. On the lower side, we have the lower epidermis, lower cuticle. Yeah. So these palisade cells, they are the second from the upper side, and you find that these ones, they have more chloroplast than the spogy mesophyll cells. That's why you find that green plants usually have the pigmentation for the green color is deep on the upper side than the lower side, simply because it has got more chloroplast, and also it do not overlap. These cells for the palisade cells do not overlap. The other one is the spogy mesophyll. The spogy mesophyll cells. These ones, they are found on the lower. These ones, they are found, that is after the palisade cells, and the ones that has got the vascular bundles. The vascular bundle that has got the xylem and also the phloem. The xylem, the function of the xylem, it is to transport water and mineral salts from the roots to the leaves, and then for the phloem, it translocates the manufactured food from these cells that you're calling the photosynthetic cells to other parts of the plant. So now we can go to the dark stage of this process of photosynthesis. We are saying that it takes place in the stroma. So this one occurs in the stroma. And what happens here? The hydrogen ions that was produced during the light stage combines. So here we have the hydrogen ions. It combines with carbon 4 oxide, and when these two combines, it is going to give us two products. That is, it is going to give us food, whatever you're calling now the food for the plants, that is glucose, going to give us glucose, and also water molecules are going to be produced. So there is water that is produced. And in this process, also the adenosine triphosphate, which is the energy that is produced in this process of photolysis, it is also used in this combination of hydrogen ions and carbon 4 oxide to produce glucose and water. And this process is known as carbon 4 oxide fixation. This one is known as carbon 4 oxide fixation. So this is generally what this process of photosynthesis entails: the light stage and the dark stage. So now from there, we are going to move to some other areas, like for example, we have just looked at mentioned about the internal. These parts, when you talk of the grana, we talk of the stroma. These are parts of a chloroplast. And these chloroplast, as you talk of the guard cells, the palisade and the spogy mesophyll cells, all of them, they are found in the internal structure of a leaf. So maybe we are going to look at the external structure of a leaf. the external structure of a leaf, the external structure of a leaf, and then so this leaf, so the leaf looks like this. There is so the leaf looks like this. Then we have this part. This is what you have, the leaf apex. We have the leaf apex. We have the leaf margin. We have the leaf margin. This is what we call the veins. The veins. Then you also have another part here that we call petiole. And lastly, we have what you call the midrib. So basically, this is an external structure of a leaf, and possibly if you should maybe go ahead and look at all those other the structure of this one is a dicot, so you should go ahead and look at the structure of a monocot, and also look at the internal structure of that leaf. For now, maybe I may look at the adaptation, one of the common question, which is the adaptation of a leaf to its functions. Adaptation of leaves to its function. When you talk of adaptation, the term adaptation, for you to get the full mark for the word adaptation. For example, we talk over, maybe give an example of one of the specialized cell in one of the specialized cells in animals, for example, a sperm cell. What are you expected to, how are you supposed to respond to this? This part on adaptation, these question on adaptation usually have three parts. So you have to mention the part here. How is it modified to do what? To do what? Any question pertaining adaptation, your answer, even if you're not going to draw in terms of, you're not going to tabulate like this, your answer should have these three parts. For example, a sperm cell. A sperm cell, we know that a sperm cell, maybe we can have an example, just like an example of a sperm cell that looks like this. A sperm cell that looks like this. So we have the nucleus, the acrosome there. We have the acrosome. We have the nucleus. This is the tail. This is the head. So what are some of the adaptations of this sperm cell? Here we have the mitochondria. When it is one is a mitochondrion. When it is one is a mitochondrion. When there are many, it is Mitochondria. So one of the adaptations of a sperm cell, we talk of having numerous mitochondria. These numerous mitochondria, they're the ones that are going to provide enough energy to give for this sperm cell to be in a position to propel into the fallopian tube for fertilization to take place. So we talk of its modification. This part is the one that matters a lot. How is it modified? Because this modification is the one that brings about efficiency. That's why we don't have a question like giving the adaptations of a microscope, because it's just but a part and its function. But for the adaptation, you must show how that organism or how that part is adapted to its functions. So for the adaptations of the leaves to photosynthesis, we are going to go through them very fast. So you find that the lamina, this is a lamina this is a lamina here. Lamina is broad. We have broad and flat lamina, and this one increases the surface area for the absorption of carbon dioxide and also the trapping of sunlight. Then we find that the epidermis is also thin, and this one reduces the distance to be covered by the gases for efficient gaseous exchange. Then you find that on the lower side, or that is where we have the spongy mesophyll cells, it usually have the large air spaces, and these large air spaces, it is for ensuring that gaseous exchange is efficient because it also stores carbon dioxide. The other one is the cuticle. The cuticle and epidermis. Those two, they are transparent, and them being transparent, they allow light to penetrate through. Then we also have the leaf mosaic, whereby you find that the leaves do not overlap. This one prevents or ensures that the plants get maximum sunlight. It exposes these photosynthetic site to the sunlight that is required for the process of photosynthesis. Then we also have the cuticle being waxy, waterproof. Waxy and waterproof. This one reduces the rate at which the plants lose water. The plants lose water. Now from there, maybe we can take some few minutes and look at. We have talked of a leaf carrying out the process of photosynthesis. How do these leaves? How do you test whether a leaf has got starch? Whether the leaf has been, whether a leaf has carried out this process of photosynthesis. Does it have the starch? Remember, when this process of photosynthesis takes place, what is formed is glucose. Glucose is formed. And this glucose, when it is formed, it is stored in plants in form of starch. That's why we test the presence of starch in a leaf. So what's the procedure of testing whether a plant, whether a leaf has got a starch? So let's look at test for starch in a leaf. Test for starch in a leaf. So how do we test whether, what is the procedure for testing whether a leaf has got starch? So the first one, you have to boil the leaf. And what is the essence of boiling? What is the essence of boiling this leaf? Because it should be put in boiling water for 10 minutes. One, it is to kill the protoplasm. The other one, The other one, the reason as to why we boil the leaf, one, it is to kill the protoplasm, and we also ensure that we denature the enzymes. Denature, it is to destroy. And why do we have to kill this protoplasm? Why do you have to denature the enzymes? This one, it is to ensure that you stop any further chemical reaction. To stop any further chemical reaction from taking place. So that is the first step. The second one, after boiling the leaf, then you remove the leaf and you put the leaf in a test tube containing alcohol or methylated spirit. Alcohol or methylated spirit. Why do you have to put this leaf in this test tube containing alcohol or methylated spirit? This one, it is to remove chlorophyll molecules. To remove the chlorophyll molecules. Then stage 3, after putting this leaf in a test tube containing alcohol, then you put this test tube in a hot water bath. Put the test tube in a hot water bath. So you put it in a hot water bath. This is to ensure that all the chlorophyll molecules has been removed. Now from there, number 4, remove now your leaf from this test tube containing alcohol or methylated spirits, and you put the leaf in warm water. The reason as to why you put this leaf in warm water, it is to soften the leaf. And lastly, now you remove your leaf in this hot water, warm water, and you put the leaf on a white tile. Then number 6, flood it. Number 6, then flood it. You flood the leaf with iodine solution. Yes. So now this is how you should carry out this process. And for you to get a positive result, at first, before even you get your leaf, the one that you're going to carry out, the one that you're going to pass through this process, ensure that if you want a positive result, if you want the results to be positive, ensure that you expose this plant, you expose the plant to sunlight for at least 6 hours before carrying out this test. You expose the plant to sunlight for about 6 hours before carrying out this process. So it is very easy, and maybe possibly if you use a diagram to show this, it's very easy. The first one, you're talking of you boiling the leaf. So the leaf is here, and we have heat. So from there we put it in a test tube. So remember this one, here we have water, here we have methylated spirit, then this methylated spirit, we're just going to have a sketch over this. So this is hot water. Then from there you you remove now your leaf and you place it to. You remove your leaf and you put it in your leaf now in hot or warm water. Should be warm water. You place it in warm water. Then from there, after that, now you have your white tile. You have your white tile there. You have your leaf, and then you flood it now. You flood it with now this one is we have now this one being iodine solution. This is iodine solution. So this one is as simple as this. Maybe a diagram. This is step number 1, step number 2, step number 3, number 4, number 5 and 6. So whereby you have to. So if you had exposed your green leaf to sunlight for 6 hours, the results here, your leaf is going to change to blue black. This one, conclusion to this is that starch is present. Just in case maybe you never exposed your plant to sunlight, maybe it was in darkness, you had destarched, you had put the plant in a dark room for around 48 hours, the leaf here is going to take up the color of iodine solution, which is brown in color, and that one shows that there is starch is absent. In this topic, there is more of these experimental questions that are very crucial in paper 2. We have the testing whether chlorophyll is required in the process of photosynthesis. There is to test whether the gas that is produced during the process of photosynthesis. There is also other experiments like physiological processes that are usually very common in paper 2. That is section A, where we have experimental questions, and each experimental questions usually have 8 marks. So there is much to cover on this. So this marks the end of our lesson. Thank you for you as my audience, and see you next time.